Good afternoon. Welcome back to Virology. Today, I want to talk about genomes and genetics. The viral genome, what it is, what it does, what it doesn't do, and how we can manipulate it. That's the genetics part. Any one of you ever heard of Erwin Chargaff? Have you heard of Chargaff's rules, right? That A pairs with T and G pairs with C, right? Did you know he was a professor at the medical school of Columbia? Probably not. When I arrived, he still had centrifuges with his name on it. He was long gone though. Erwin Chargaff. So that's the quote from him on this slide. So in the 1950s, we have a breakthrough in understanding viruses. We find out that the viral genetic information, the nucleic acid, is the genetic code. Now, we had already figured this out in 1944 for bacteria. In 1944, famous experiment, Oswald, McLeod, and McCarty here in New York City showed that bacterial DNA was the genetic material. And then in the 1950s, it followed for viruses. And there are two experiments I'll briefly go through with you that show that for DNA and RNA viruses. Remember, viruses can have DNA or RNA as the genetic code. So we're going to talk about Hershey-Chase's experiment with phage, bacteriophage T2, and Frankel-Conrad's work with tobacco mosaic virus. So the Hershey-Chase experiment, you probably learned in high school and already forgot. And I will remind you, and then once you're done here, you'll forget again. But you'll know where to look for it because you will have all these lecture videos and notes. So the Hershey-Chase experiment uses a kitchen blender. And if you go to Cold Spring Harbor out on Long Island, the laboratory, they have a library. And in the library, there is the kitchen blender that Hershey used. So there's Hershey, Al Hershey, and his technician, Martha Chase. They did this experiment in 1952. What they did was they want to know, is it the protein or the nucleic acid that's the genetic information? So they prepared phages radioactively labeled with either sulfur or phosphorus. You could buy radioactive sulfur or phosphorus, and you can grow the phage in the presence of either. So the sulfur, of course, will label the protein, and the phosphorus will label the DNA. Then you infect cells with these viruses. You allow the viruses to attach, and you just give them 5 minutes to attach, and then you put them in a blender and shear off any phages, and that prevents any more infection from happening. That's the use of the blender. Personally, I can't imagine putting radioactive stuff in a blender. Do you know what kind of aerosol that makes? Huge. But that was the 50s. We didn't really think about safety back then. So you incubate the infected cells, and then you look for the production of new phage, and you ask, where is the radioactive label? And the phage, they found no sulfur in the phage, and they found phosphorus, P32, in the progeny phage. So the DNA is labeled, and that proved that DNA, so because DNA is labeled, it gives rise to new phages, some of which contain radioactive label in them. So that proved that DNA was the genetic material. Similar experiment without radioactivity done for tobacco mosaic virus. Now, this is a virus that infects plants. And early on, Frankel and Conrad, it's one person actually, Heinz Frankel Conrad, he found that, we know that the virus infects plants. He found that you could purify the coat protein and the RNA. So this is a virus that has RNA genome. The RNA is shown as a coil here, a helix. And the virus particle is made up of this RNA complexed with many subunits of the coat protein. Coat protein is a single protein we'll talk about next time that's repeated many times that protects the RNA. And that's the virus particle right there. So he had two different strains of tobacco mosaic virus, which he could distinguish. you could distinguish the proteins of these strains. And we'll call them green and red for simplicity here. So what he did is he purified the green RNA and the red RNA, and he purified the red protein and the green RNA and mixed them together. So he mixed red protein with green RNA and green protein with red RNA. He assembled particles. So now you have a mixed virus. You have a green RNA with a red protein, for example. You infect plants and you see what comes out. And the coat protein always matches the genome. The coat protein, if you have a green genome, the coat protein is green. If you have a red genome, it's red. So again, the RNA specifies the genetic material, not the protein itself. So those are two really important experiments that showed the nature of the genetic information. But the bigger surprise was that even though, and as I told you last time, there are billions and billions, to channel Carl Sagan, billions and billions of viruses out there. Maybe you're all too young for Carl Sagan, I don't know. But he used to get on television and say, the universe is made of billions and billions of stars. So we have billions and billions of virus particles. But it turns out there are only 7 different kinds of virus genome. Number 7, you can easily remember that, the number 7 train, right? And even easier for you, all viruses have to make mRNA that can be read by host ribosomes, because viruses do not encode a protein synthesizing machinery. All viruses on the planet do this, as far as we know. They have to make mRNA that can be recognized by host ribosomes. So that will help us to arrange these 7 viral genomes by a unique pattern. And this is it, the Baltimore scheme. So this is a scheme developed in the 1960s by David Baltimore, who's shown up here at the upper left. We're going to come back to him later because besides him doing this, he also co-discovered the very important enzyme reverse transcriptase. But he used this insight that every virus has to make mRNA to organize the 7 viral genomes. And this is the scheme he made. He put mRNA in the middle. Why? Because all virus genomes have to make mRNA. And then he put the viral genome types around it, showing the pathway to mRNA. He actually didn't have the 7th group because they weren't known at the time. These were the hepatitis B viruses, which have a very convoluted scheme, which we'll come back to in a minute. So class 1 are viruses with double-stranded DNA. Those viruses can make mRNA directly from the DNA. You have to remember a few things for this to work. The first is that the synthesis of mRNA from DNA can only happen with a double-stranded DNA template. It won't happen with single strand, it won't happen with a gapped double strand. But the double-stranded DNA can go right to mRNA. Group 2 are viruses with single-stranded DNA. Those have to be copied to make double strand before they can get to mRNA. It's just a fact that you'll have to remember. Group 3 now, group 7 are the viruses with also double-stranded DNA genomes, but they're gapped. They have spaces missing. They have parts missing, and we'll get back to that later, why that is. But these viruses have to, and that cannot be transcribed into mRNA. So it has to be copied to DNA, which could then be copied into mRNA. But those viruses actually reverse transcribe the RNA into single and then double-stranded DNA, which eventually leads to mRNA. So that's the DNA viruses, 3 different classes. Then we have RNA viruses, class 3, double-stranded RNA, both strands in a helix, a double helix. That virus or genome cannot be directly translated because even though there's a plus strand, a messenger RNA strand in that double strand, the ribosomes can't access it. So the minus strand has to be copied to make mRNAs. So you have to remember that double-stranded RNA can't be translated. It's group 3. Group 4 are plus strand RNAs, probably the simplest of all. The plus strand in these viruses is translated into protein. And to make more plus strands, you have a negative strand intermediate. Group 5 is negative stranded RNA viruses. So these cannot be translated to the complement of mRNA. And so they have to be copied to make mRNA. And then group 6, viruses which also have a plus strand RNA, but it is not translated. It is instead copied in the cell by reverse transcriptase, this very special enzyme to make a DNA copy that then gives rise to mRNA. So that's the scheme, and it's really about understanding the pathway to mRNA for these 7 different genomes. Now, a couple of definitions before we go further. First, we use plus and minus in a very specific way. mRNA is the plus strand, and it's just a convention. It has nothing to do with electrical charge or anything else. It's just, that's the convention. And so the DNA of equivalent polarity would be called the plus strand as well. RNA and DNA complements are the negative strands. So the complement of mRNA would be a minus strand. And remember, as I've just alluded to before, not all plus RNA is mRNA. And what I mean by that is just because an RNA is plus stranded does not mean it's going to be translated. An mRNA is something that is translated into protein. So the elegance of the Baltimore scheme, it's really about the pathway to mRNA. Knowing only the genome structure, you should be able to tell me, like I just did for you in the previous slide, the basic steps that occur to get to mRNA. For example, knowing that double-stranded DNA can be directly transcribed and that double-stranded RNA cannot be translated and so forth. So there are 7 classes of viral genomes. We have 3 double-stranded DNA genomes. We have double-stranded DNA class 1, double-stranded DNA with reverse transcriptase, that's class 7, and then we have single-stranded DNA, and then the RNA genomes, double-stranded RNA, and then 3 single-stranded RNA genomes of plus or minus polarity, and another with reverse transcriptase. So RT is special. It makes a genome different from all the others. That's why we have a DNA and an RNA virus class with reverse transcriptase. Okay, so let's take a quiz to see if you got that. So the first question is, why is mRNA placed at the center of the Baltimore scheme? Because all virus particles contain mRNA. No reason, because all viral genomes are mRNAs, because mRNA must be made from viral genomes, because Baltimore studied mRNA. Okay, let's have a look how you did. Most of you got D, because RNA must be made from all viral genomes. That's the right answer. Because all virus particles contain mRNA. No, we know that's not true, right? The Baltimore scheme. How many genomes are there? 7, right? Number 7 subway. I know none of you probably ever take the 7 subway, but that's the best I could do. Because all viral genomes are mRNAs, they're not. Because Baltimore studied mRNA, no. Because mRNA must be made from all viral genomes. Now, in addition to these 7 different classes that I just showed you, RNA and DNA genomes are really diverse. They come in all different configurations. As you can see here, we have linear, double-stranded, we have circular, double-stranded, we have also double-stranded RNA, although it's actually single-stranded and all base-paired within itself. Segmented genomes, which are in pieces, the gapped genome of hepatitis B virus. We have single-stranded plus and minus. We also have what's called ambisense. It's got a little bit of plus and a little bit of minus. We have double-stranded. Some genomes have proteins attached to them. Some genomes have the ends covalently linked in this double-stranded genome here, and some of them have an attached piece of RNA. Now, the colors I use are all consistent. I have a color scheme, so RNA is always green and DNA is blue. So here's RNA, And RNA can be two shades of green. It can be like this Kelly green here, or it can be an olive green, right? It's not brown, it's olive green. So that's RNA. And then DNA can be blue. The dark blue is the plus strand, and the light blue is the minus strand. And proteins get other kinds of, like earthy colors. That's what the artist said, they're earthy colors, okay? So I would know this, even though when you take your exam, you're going to be allowed to have a sheet of paper with stuff on the front and back of your choice. But I would still learn this because you could copy it onto your sheet. That's fine. You can make a really small version, but you still should understand it, right? What does it mean? Because you could look at it during the exam and say, I have no idea what this means. And then you're in trouble because you only have 75 minutes. So I would go through this and know what it means, because if I tell you a genome, you're going to know how to get to mRNA right away. That's the beauty of this. Now, in this class, we only talk about certain viruses, and they're shown here by their genomes, like double-stranded DNA viruses. We're going to talk about adenovirus and herpes simplex virus. We're going to talk about hepatitis B virus for the class 7, parvoviruses for the class 2. So the virus, if you have a dog or a cat, you need to immunize them against that virus. The double-stranded reoviruses, influenza viruses are negative-stranded, polioviruses plus-stranded, and then the retroviruses like HIV-1 are having the reverse transcriptase. So what's encoded in a genome, now that we know what they look like? Well, a lot of things are encoded. We have gene products and signals. So the genome encodes proteins, but there are also non-coding regions of the genome, which are typically regulatory signals. And so there are some viruses that encode parts of the protein synthesis machinery. No virus encodes it all. There are proteins and signals for replicating the genome. There are proteins for assembly and packaging the genome into a particle. There are proteins that regulate the infectious cycle and time it. There are proteins that modulate host defenses. Talk about those in the second part of the course. And there are proteins that enable viruses to spread to other hosts. So lots of different functions are involved here. I want to tell you about one specific giant virus. Remember we talked last time briefly about, we used to think all viruses were pretty small, and now we've got big ones. And this is one called Tupan virus, isolated down in Brazil. And there's a reconstruction of the particle, just beautiful. This one encodes almost the complete protein synthesis machinery. Only the ribosome is missing. It doesn't encode the ribosome. It encodes 20 amino-acyl tRNA synthetases, right? The enzymes that put amino acids onto tRNAs. It encodes 70 tRNAs, multiple translation, initiation, and elongation proteins, a lot of genes related for the maturation of tRNAs and mRNAs. It is the most complete translational apparatus of the virosphere. Virosphere being all the viruses on Earth. And you may ask, what's the function of this? And we're not sure because nobody has done the right experiments yet. But, you know, codon usages differ in cells and in viruses. Just in different organisms, they're different. So a yeast is different from E. coli from us. And it's the same for viruses. So we think maybe by producing tRNAs that the virus can replicate faster or more efficiently, but we really don't understand why this is the case. It's mostly the giant viruses that are like this. What's not in a virus genome? This actually changes from year to year. There are no genes encoding the complete protein synthesis machinery, so ribosomes are not encoded, so that would include the ribosomal proteins and the ribosomal RNAs. I used to say there are no genes encoding proteins involved in membrane biosynthesis. That's out the window because now viruses were found that do have some of those. No classical centromeres or telomeres that we find in standard host chromosomes. So our chromosomes have centromeres and telomeres. We don't see those in viruses. But I would say, as I teach this course from year to year, some of these may go because when we sequence new giant virus genomes, 90% of what we see is novel. We can't find the proteins in any database. And so we're always finding new things. I thought you might like to know the long and the short of things. What are the longest viral genomes and the shortest? So these are the largest virus genomes. They're all DNA viruses. And that's a trend because DNA is way more stable than RNA, and it can get longer. So here, the biggest virus genome so far is Pandora virus. There are two different isolates. The biggest one is 2.4 million base pairs of double-stranded DNA, encoding 2,500 proteins. And most of those proteins, we have no idea what they do. Because if we look at the sequence, you can use computer programs to take the sequence and compare it to all the known proteins. It doesn't match up with anything. So a lot of dark matter in there. And then there's another one, 1.9 million, and it goes all the way down. There's Tupan virus, which we just talked about, 1.5 million base pairs, 1,400 proteins, and then all of these other viruses with interesting names, down to 600,000 base pairs. By the way, the genome of Haemophilus influenzae, which is a bacterium, is 1.8 million bases long. So some of these viruses have longer genomes than a free-living bacterium. And down at the lower right, Nasua is a parasitic bacterium. It lives inside another bacterium, and that is 112,000 base pairs in length. Now, just to explain some of this naming, it's actually pretty quirky. So the first giant virus was called virus. And it was discovered in France. And the French have a sense of humor. To this day, they still do, despite everything that's happened. And so when they discovered the next one, they decided to call it Mama virus. And when they found the next one, they called it Mumu virus. And when they found the next one, they called it Mega virus. So that's because they have a sense of humor, and I appreciate that. There's no meaning. Mimi has a meaning. Mimi means microbe mimic, because this virus was first discovered in a patient who had an infection, and it was thought to be a bacterium because they could see it under the microscope, under the light microscope. So they actually put it away. They froze it, and nobody paid any attention to it for 10 years. And then someone took it out of the freezer and said, hey, this is not a bacteria, this is a virus. So that's why they called it Mimi virus. The largest RNA virus genomes pale in comparison. The biggest one, 41,000 bases. It's a single-stranded plus RNA. It's a virus that infects planaria. Remember those worms that you may have played with in 5th grade, right? They have viruses like everything else. Then we have an Aplysia virus, 35 kb, and a snake virus, a python virus, 33 kb in length. These are all plus-stranded RNA genomes. The smallest genomes are shown here. The smallest genome is 220 bases of RNA. 220 bases. Can you imagine? It's tiny. It doesn't encode any protein, actually. It actually needs another virus to help it to reproduce, and we'll talk about that later on. Then we have viruses that encode protein. We have hepatitis delta virus, 1,700 bases encoding one protein. Then we have some DNA viruses that are pretty small, circoviruses and enteroviruses. By the way, these circoviruses and enteroviruses, they infect all of us. If you take blood, everybody's blood has enteroviruses in them. And apparently they don't harm us. And then all the way down to 4,000. So you see not a lot of proteins here, not very long compared to the DNA viruses, but obviously they all work. They can all function. They can get into cells and redirect the cells to make virus genomes. Okay, the next question is, what information may be encoded in a virus genome? Complete protein synthesis systems, centromeres or telomeres, enzymes to replicate the viral genome, or all of the above. Okay, let's see what we did. Most of you got C, the right answer. enzymes to replicate the viral genome. They don't encode complete protein synthesis machinery, right? Remember, only the ribosome is missing in the two panviruses. No centromeres or telomeres, and not all of the above. I'll just show you the slide that has this. What is encoded, right? Replication of the genome, what is not encoded, so that was the answer. No centromeres or telomeres, no genes encoding the complete protein synthesis machinery. Now let's talk about viral DNA genomes first. These viruses with double-stranded DNA predominate in the bacterial virosphere. That is, all the viruses on Earth that infect bacteria. And many viruses emulate the host in terms of how the DNA is manipulated. We'll talk about that in another lecture. But most viral DNAs are not like cell chromosomes. A few are, and I'll point out those exceptions. And they do different things compared to the host. And there at the bottom, that picture is that's a picture of chromatin, right? DNA wrapped around histones, and that's the way the DNA exists in cells. And most DNA viruses, it does not look like that. It's not chromatinized, as we would say. So here are some double-stranded DNA genomes. Again, the double-stranded DNA can be transcribed to form messenger RNA, which then is translated into proteins, and the DNA is replicated to make new virus particles. And we divide these into two classes. On the left, genomes that are copied by host DNA polymerase. So our cells, as you know, surely, have DNA polymerases to replicate our genome and RNA polymerases to make the various RNAs that we have, tRNAs, ribosomal, et cetera. These viruses are too small. The genome is too small to encode a DNA polymerase. So it utilizes the DNA polymerase of the host cell. An example is a polyomavirus with a 5,000 base pair circular double-stranded DNA. We'll talk about those a lot. And then on the right, we have virus genomes that encode DNA polymerase. These are larger viruses. They have a lot of coding capacity, so they can do that. Adenoviruses are between 30 and 50,000 base pairs, double-stranded DNA. Herpes viruses between 120 and 240, and the pox viruses even bigger. And of course, the giant viruses as well. I'm not including them here, but certainly they have genomes of some of them over 1 million base pairs, so they can encode a DNA polymerase. Now, examples of some of these viruses are shown here that have double-stranded DNA virus genomes. We have adenoviruses here. This will be one of the viruses we talk about in some detail in this course. All of these, by the way, cause human disease except pithovirus, which is a giant virus. The pithovirus shown here, you can see, is much bigger than any of the others. It's not even drawn to scale. It won't fit on this slide if I drew it to scale, but that's the double-stranded DNA genome. Herpes viruses, enveloped viruses with glycoproteins in the envelope, and then some more non-enveloped viruses, the papillomaviruses and the polyomaviruses, and then finally the pox viruses like monkeypox virus and smallpox virus. So those are examples of some double-stranded DNA-containing viruses, and we'll talk about these later on. How about those DNA viruses with reverse transcriptase? That's group 7. So these are the hepatitis B viruses, which have been in the news lately because the wise CDC has just taken away the birth dose of hepatitis B vaccine. We'll talk about that later. So this is a virus with a gapped double-stranded DNA genome. It has to be repaired, and then it can be transcribed to mRNA, which is made into protein, of course. But some of that mRNA is reverse transcribed to DNA, double-stranded DNA, and it's packaged in the virus particle. And this is what the genome looks like. It is gapped. It has a partially double-stranded character. It also has a protein attached and a piece of RNA. So this genome cannot be made into mRNA. It has to be repaired. It has to be double-stranded, and we have to take off the protein and the RNA. Why they're there, we'll find out later when we talk about how this virus reproduces. Now we have viruses with single-stranded DNA genomes. Again, the single strand cannot be translated. It has to be made into double-stranded DNA. These viruses can package either the plus or the minus strand. It doesn't matter because when either gets into cells, it's made double-stranded. And then the double-stranded, of course, can be made into message, which gets made into protein. And here are two examples of viruses with single-stranded DNA genomes. The circle viruses, between 1.8 and 2.3 kb. These are single-stranded circles of DNA in the virus particle, of course. An example is TT virus, which again is in all of us. It's in everyone's blood. In fact, when you donate blood, it's got this and other viruses in it, and we cannot reject the blood based on the presence of these viruses because we wouldn't have a blood supply. So there doesn't seem to be any pathogenic consequence of this. And on the right are the parvoviruses. And remember, these are viruses that infect both humans and animals, non-human animals. This one is an example of V19 parvovirus, which causes in children what's called fifth disease. So there's 6 childhood rash diseases, and this is one of them, the fifth one. The DNA is very unusual. It's single-stranded, but has these terminal repeat sequences. You can see A and A prime are complementary at each end, and they form these hairpins at either end. And the significance of that we'll get back to in a little bit. Our next question is, which DNA genome on entry in the cell can be immediately copied into mRNA? Double-stranded DNA, gapped double-stranded DNA, circular single-stranded DNA, linear single-stranded DNA, all of the above. Hey, let's see how you did. Most of you got double-stranded DNA. That's the first answer. That's correct. Only double-stranded DNA can be immediately copied into mRNA. The others have to be fixed first, right? The gap DNA has to be repaired. The linear single-stranded has to be made double-stranded. So that's the only one that can be directly transcribed into mRNA. All right, let's talk about viruses with RNA genomes. These are the best. Look, on my backpack, I have an RNA molecule because they're the greatest viruses on the planet. Well, I used to work on an RNA virus. That's why I have that. RNA genomes dominate the eukaryotic virus sphere. So there are some in bacteria, but way fewer than in eukaryotes. And here is a little picture showing you the abundance of different kinds of RNA viruses. We have plus RNA, double-stranded RNA, and minus RNA here. And you can see this chart, this bar graph on the right shows you the number of families in plants, invertebrates, bacteria, archaea, and others. So these plus, you can see the bacteria for the plus, the double-stranded, and the minus RNA are vanishingly rare. We are discovering more recently, more than shown here, but much different from in plants and invertebrates. Now, for RNA viruses, the cell cannot copy the RNA. Cells can copy DNA because they have a DNA polymerase. But given RNA from a virus, a cell cannot copy it. Consequently, every virus genome on the planet that has an RNA genome has to have an RNA-dependent RNA polymerase to copy it. So mammalian cells don't have that enzyme, so I will abbreviate it as RDRP. And these genomes of all these RNA viruses have to encode it or use it from another virus, as you'll see later. So these RNA-dependent RNA polymerases not only copy the genome, in other words, replicate it, but they also copy it to form mRNA. So they do both things. Let's look at some examples. Here are the double-stranded RNA viruses. The example here, again, remember the double-stranded RNA cannot be translated directly into protein. It has to be copied by the viral RNA polymerase to make mRNA. So that plus strand in there is not accessible to the ribosome. So the first step is synthesis of mRNA, then it can be made into proteins. And so a very important virus with double-stranded RNA are the rotaviruses, causative agents of human gastroenteritis. We used to have, this is mostly happening in very young kids. And we used to have 70,000 cases a year in the US of gastroenteritis in young children caused by rotavirus. And the use of the rotavirus vaccine has completely eliminated that. These viruses have double-stranded RNAs. This one in the reoviruses has, this the family reoviruses have 3, 6, 7, 8, 9, 10 double-stranded RNA segments, but the number of segments varies according to which virus we're talking about. So, okay, that's the reoviruses. And we have viruses with single-stranded RNA, and we have a number of different families here. We have the coronaviridae, or the coronaviruses, which you should recognize. SARS-CoV-2 and SARS-CoV are both coronaviruses. So they have very long genomes, up to 41 kb, plus-stranded RNA. Typically have a cap at the 5' end. We'll talk about what that does, and a poly-A at the 3' end. Flaviviruses, West Nile virus, dengue virus, yellow fever virus, they are plus-stranded. Picornaviruses, polioviruses, and rhinoviruses, and finally, togaviruses, more mosquito-borne viruses. These RNAs, again, are unique because they can be translated directly into protein. So the RNA gets into the cell, it's translated immediately, and then if you need to make more genomes to put in virus particles, you simply make a minus RNA intermediate. And that's a process we'll talk about in some detail later on. So here are some examples of RNA pathogens with plus-sense genomes. We have the picornaviruses, as I said, poliovirus and rhinoviruses. Poliovirus has been pretty much eliminated in the US and many other countries by vaccination. The new CDC says, we don't have to do this. It's your choice whether you should protect your kid against polio or not. Really good. Caliciviruses causing gastroenteritis, like noroviruses. Go on a cruise ship, you vomit for 2 days, 3 days, vomiting and diarrhea. That's norovirus. We'll talk about that later. Then we have the coronaviruses. So the picornas and the calicis are viruses made of pure protein, no envelopes, and the coronaviruses have an envelope. And whenever there's a membrane around the particle, then you have to have glycoprotein spikes in the membrane so the virus can attach to the cells. So that's coronaviruses. Flaviviruses, also envelope viruses, and you can see some examples of them there. And then togaviruses, rubella virus, and equine encephalitis virus. So rubella virus used to infect many people in the US. And if you got infected when you were pregnant, you had a very good chance of having a child with congenital birth defects. There were many thousands of kids who were blind or deaf or had other issues because their moms got infected with rubella virus. So what do? We developed a vaccine. Now there's no rubella virus in the US. And so again, a guy at the CDC said, I never see any congenital rubella. Why do we need the vaccine? This is what we're stuck with, folks. All right, retroviruses plus RNA with DNA intermediates. This is the Retroviridae family, and there are 2 human pathogens in this family, HIV and HTLV. And we will have half a lecture on HIV later on, of course, the causative agent of AIDS. Now, these viruses have a really interesting reproduction strategy. They have a plus RNA genome, but when that gets in the cell, it is not translated into protein, despite it being plus-stranded. So beware of plus-strand RNAs. They're not all messenger RNAs. This one is copied into DNA, first single and then double-stranded, by reverse transcriptase, an enzyme that's present in the virus particle. Then that DNA goes into the nucleus of the cell. It gets integrated into our chromosome, and there it's transcribed into mRNAs. And that's the genome there, 7 to 11 kb of plus-stranded RNA. And all of us, as I said on day one, about 8% of our genome consists of integrated copies of old retrovirus infections. We have them in us, and the consequences of that we will talk about. Viruses with negative strand RNA, a lot of pathogens here And again, negative stranded RNA can't be translated. That's the definition of a negative strand. So that has to be copied into plus strand RNA when it comes into cells, which can be a messenger RNA, and then it can be translated to proteins. So what copies it? The RNA-dependent RNA polymerase that's in the virus particle, and that's encoded by the virus genome. Now, plus strand RNA viruses do not have to have the RDRP in the particle, because when the RNA gets into cells, it's immediately translated. But negative strand viruses, if the RNA gets in the cell, nothing will happen. So they have to bring in a polymerase with it. So all these negative strand RNA viruses contain a polymerase. These viruses can have a genome in pieces. They're called segmented. So for example, the influenza viruses have a 8-segment genome. And there are also viruses with non-segmented genomes like measles viruses and rabies virus, Ebola virus, if you will, all negative sense genomes. So here are some examples of viruses with negative sense genomes. We have paramyxoviridae, which include measles and mumps viruses, rhabdoviruses, which include rabies virus, the filoviruses, Ebola and Marburg viruses, influenza virus, and the arenaviruses, which include Lassa virus. And I just want to point you to this book called Fever, written by John Fuller. It was written in the 60s. So when I graduated from college, I didn't know what to do with my life, right? College didn't help me decide. So I got a job in a lab, and I found this book, and I read it, and I said, I have to be a virologist. So that's why I'm here today, because of this book. So maybe if you read it, you'll get inspired too. It's about the first emerging virus that we learned of, Lassa virus, which still causes problems to this day. Now, when you have a segmented genome, that is a genome in pieces, like influenza virus has 8 segments, it can do something called reassortment. Okay, this is going to be very important later on when we talk about the epidemiology of influenza viruses. So here we have 2 different influenza viruses infecting one cell. And that happens often. Cells get co-infected by multiple viruses. And these 2 viruses are distinguishable. They have different RNA genomes. But they're both influenza viruses. They go in the cell, the genome replicates, and when new viruses are made, these new viruses have 8 segments from this pool of RNA in the cell, and they can have segments from the original virus, one of the original viruses, the red here, or the blue, or sometimes they have a mixture of red and blue segments. That's called a reassortant, because the segments are reassorting in the cell. And this is a very nice way to get new virus particles. And so the consequence of that is big for influenza virus. It leads to pandemics when reassortants occur. We'll talk about that later, of course. And finally, we have our ambisense RNA genomes, which are a mixture of plus and minus polarity. As you can see here, they 2 different color greens. And these are arenaviruses and the Peribuniaviridae. They have 3 different sized RNAs in the virus particle. And the particle contains RNA polymerase. So that tells you that despite these viruses having some plus sense, the first thing that happens when they get into cells is not translation, but synthesis of an mRNA. And that's why you need the RNA polymerase in there. But then this part that is plus stranded can be translated later on in infection. Now RNAs, all the RNAs I've shown you so far, I'm just showing you lines on the screen, right? But RNAs do not exist as lines. They are highly structured molecules, which are shown here. So here's a typical picornavirus genome that I've been showing you on the upper left, a line, a squiggly green line, but that's not how it looks like in real life. On the bottom is more like it, but even this is not accurate. There are extensive secondary structures in this RNA. So RNAs can form these stem loops because they're complementary sequences along the stretch of the RNA. They can be simple, or they can be very extensive with multiple branch points, as you can see here. and they can interact with each other. So this red arrow shows that these two red sequences actually base pair in the virus particle in the RNA. And so they're very extensive secondary and tertiary structures of RNA. And they all have functions. They're not there just because they're nice, but they help the virus to reproduce. Here's another example in the leader of HIV-1 RNA, bunch of stem loops. And you can see drawing them in this way is actually misleading because they form 3D structures like those shown here. Okay, our last one for today is what statement about viral RNA genomes is correct? Plus, single-stranded RNA genomes may be translated to make protein. Double-stranded RNA genomes can be directly translated to make protein. Plus, single-strand RNA virus replication cycles do not require a minus-strand intermediate. D, RNA genomes can be copied by host cell RNA-dependent RNA polymerases, or all of the above. Okay, let's see how we did. Well, only half of you got the right answer, so I didn't explain something, I guess. So plus genomes may be translated to make protein. That is absolutely correct, right? Plus may be, the key is may, right? Not always, but may, because we know the retrovirus plus strand genomes are not translated into protein. But then many of you said double-stranded genomes can be directly translated. No, they can't. I think I said that 3 times. They can't be because the double-stranded makes the RNA inaccessible to ribosomes. Then you say plus virus replication doesn't require a minus strand. It was on multiple slides that to get from plus to more plus, you need to go through a minus intermediate. And then finally, RNA genomes can be copied by host cell RNA polymerase. There is no such animal. Let me show you the slide that shows that, that says that. Mammalian cells do not have RNA-dependent RNA polymerase. So the virus has to have it itself. But anyway, you can see, let's find one here. Here's a plus sense RNA going through a minus strand intermediate to make more plus RNAs. So those are the answers there. Okay, the remainder of time, I want to talk about genetics now. We've done the genomes part. And here we have the technique, the plaque assay, that allowed the first genetic analysis of viruses, because you could take the virus from a single plaque and study it. So if you took a tube of virus and, say, treated it with a mutagen like UV light, you could then make a plaque assay of that and pick out viruses with different kinds of mutations in them. But today we do something differently, as you see. Now let me give you some terminology. A mutation is a change in DNA or RNA. It can be an addition of a base, a deletion, a rearrangement. It's manipulation of nucleic acid. And mutations can lead to amino acid changes. They can be substitutions, additions, truncations, and proteins. And in the old days, we used to make mutants of viruses by UV or by chemical mutagens. All right, so that's the definition of mutation. And today, we introduce mutations into viruses by modifying a DNA copy of the genome. This is called the infectious DNA clone. And we introduce this into cells by a process called transfection. It's actually a validation of Hershey-Chase, right? Because the DNA is put in cells and out comes viruses. And you can take that DNA and make deletion, insertion, substitution, nonsense, missense mutations. You can even make viral vectors. You can make viruses make people healthy again. Viruses can do this, can cure cancers, genetic diseases, and make vaccines. And we'll talk about that in the last lecture of this course. Now, transfection is the production of infectious virus after you put DNA into cells by a process called transformation. It was first done with a bacteriophage called Lambda. And the word comes from, it's a portmanteau, right? It's a fusion of 2 words, transformation and infection. That's where transfection comes from. So you transform DNA, or you introduce DNA into cells by transformation, and then you're actually infecting the cells. And so here's a picture of that. You can take RNA or DNA and introduce it into cells by transfection. That initiates an infectious cycle, and out come viruses. Now, of course, if you have DNA, that's beautiful because you can manipulate it. You can change it, but you can't do that with RNA. So for the first infectious DNA of an animal virus was that of poliovirus. So poliovirus is a plus RNA containing virus. If you infect cells, of course, out comes poliovirus. If you extract the RNA and introduce it into cells by transfection, it initiates an infectious cycle because it's plus-stranded, and as soon as the plus strand gets in, all the proteins are made that you need to replicate the virus. And if you take the RNA and make a DNA copy, a double-stranded DNA copy, and put it in a plasmid, a bacterial plasmid, and introduce that into cells, that will also initiate an infectious cycle, as will if you make RNA in vitro from this DNA and put that in cells. All of these lead to infections. Now, for every virus that is studied, you have a DNA copy of the genome that you can manipulate. We'll see examples of this as we go through this course. So, for example, another example of infectious viral DNA is for influenza virus, and I want to show you this because it's different from poliovirus. Poliovirus, the genome is a single strand of RNA, but influenza virus is segmented. There are 8 RNA segments. So what you have to do is you have to make a plasmid that has 2 promoters on it, a Pol2 and a Pol1 promoter. These are promoters for cellular RNA polymerases. And so these inserted into these plasmids are the viral DNA sequences, 1 for each of the 8 segments. And then when you put these in cells, you make both mRNA and viral RNA for incorporation into new particles. And so in practice, what you do is you take 8 plasmids, each encoding 1 of the genome segments. You put those in cells, and out comes influenza virus. So a little different because you need 8 plasmids, but still it works. Now, this technique was used to recover the 1918 influenza virus. So back in 1918, there was a pandemic of an influenza virus, killed millions of people, way more than the COVID pandemic. We didn't have virus in 1918. We didn't isolate influenza virus until 1933. So we didn't have that 1918 virus to study. So it would be useful to study it and understand why it killed so many people, right? So what was done in 2005, and this, for example, is a picture from 1918 where armories throughout the world were set up as makeshift hospitals for people with influenza because hospitals couldn't accommodate them. In 2005, an investigator, Jeff Taubenberger and others at NIH, they extracted influenza virus RNA from paraffin-embedded lung tissues. So as you know, 1918 was around World War I, and a lot of U.S. service people died of influenza. And so the Army took pieces of their lungs and embedded them in paraffin to make sections to look at them. But they were all stored. And so Taubenberger went and got some of those extracted RNA, and he determined the sequence of the influenza segments. In fact, I interviewed him a couple of years ago, and he showed me one of these blocks, and you could see it in the interview. He also got some samples from people who had died up in Alaska, who had remained frozen since 1918. And he was given permission to take a biopsy of their lung, and he got more RNA sequence from that. He was able to assemble the complete sequence of all 8 segments, so he made 8 plasmids, put them in cells, and out comes 1918 influenza virus. So we have now that virus. It's a very high-level containment virus. You can't work on it just anywhere. It has to be in a very high containment laboratory called the BSL-4, which we'll talk about later. Now, I've given you some examples of cloning viral genomes in plasmids, but you don't always have to use plasmids. You can actually just synthesize DNA and put it into cells. So here's an example of the recovery of horsepox virus. So this is the pathogen of horses. and probably cows as well, but not people. And it's extinct. But a number of years ago, a company decided it might be useful as a vector. So we had the sequence of the genome of horsepox virus, 212,000 base pairs. And so this company chemically synthesized 10 overlapping DNAs that made up that 212,000 base pair sequence. So there are companies that will make DNAs for you. You just type it in and they will make it. But if you type in smallpox virus, they will not make it for you, right? Because there's a screening done to make sure you're not trying to do bad things. Anyway, they took these 10 DNAs, they put them in cells, and out came virus. And this is a diagram of the genome and the 10 different RNAs. They put a yellow fluorescent protein in here so they could see that it was being produced. And they got infectious virus, and they recovered the virus to work on it. Now, yes? Not that we know of, but, you know, bona fide virologists are not going to do that. They're not going to do the wrong thing. So the idea would be a terrorist would want to do that, but usually they don't know what they're doing, right? So I don't know. We have really strict regulations here in the US, but not every country is equal in that manner either. So people are worried about it. That's why we stockpile smallpox vaccine and smallpox antivirals in case someone decides to recover it and send some infected person into New York City, because that would work. So right now, I don't know of any example, and that's why this slide has to do with, because after these examples that I've told you, a number of people got upset because this is a field called synthetic virology. You can now do experiments that weren't possible before. You can reassemble virus genomes using these DNAs, and so people started to get worried about it. For example, here's an article that described the horsepox experiment that I just told you. Scientists bring back extinct horsepox virus in the lab, raising important biosecurity questions. Well, I mean, horsepox is not a human pathogen, but yeah, you could do this with smallpox, but you wouldn't be allowed to, right? If you tried to synthesize it, you would get flagged, and FBI would come to your lab and investigate you. Now, this could happen in another country, because, as I said, we don't know what happens in other countries. So anyway, in the US, we have a committee, NSABB. It's a federal advisory committee to provide guidance on research. So when you write a research proposal, you have good goals in mind, but sometimes the results can have dual use. They could be used nefariously. And so that's what this is, dual use research of concern. This committee will say, you know, this is a good goal, but, you know, it could also be used by a terrorist. So you have to be very careful about it. And so this leads to the last thing I want to talk about, which is a hot topic now politically, and that's gain of function research. Now, scientists have been doing gain of function research for years. It is simply giving a property, a new property to an organism. It could be a bacteria, a yeast, a virus, a plant, anything. Give a new property. So, you know, you make beer with yeast. You could modify the yeast to do a better beer, and that's a gain of function for the yeast. For viruses, we can do it by modifying DNA, right, and getting virus after transfection. Or in the old days, we used to passage viruses. Let's say we want to adapt poliovirus to mice. You can pass it from mouse to mouse until you select for a mutant. That's gain of function the old way. So two ways you could do gain of function: the new way by modifying DNAs, or the old way by passaging virus. Now, here are two examples of gain of function research. On the left is a paper from 1939, I think. Transfer of the strain of polio from cotton rat to the white mouse. So we're passaging the virus in mice, and eventually it reproduces in mice. So now we've given a new property to poliovirus. It's a gain of function experiment. And I'm telling you this because it's been done for years, and the goal is to enhance our understanding of, in this case, viruses. On the right is a more recent experiment. This is actually a very contentious experiment that was being done in Wuhan, China after SARS-CoV in 2003. So the laboratory in Wuhan was trying to understand, are there other coronaviruses in the wild that are dangerous potentially to people? So they made recombinant viruses. They would isolate coronaviruses from bats in the countryside, get the sequence of the spike protein, and then insert that spike into a laboratory coronavirus and see what it does. Does it increase pathogenicity? Does it allow infection of human cells? Just to get some information on whether we should worry about it or not. And this is a growth curve of some of these recombinant viruses in mice. And what we're looking at is body weight loss on the y-axis with day post-infection. And you can see the control virus doesn't really cause weight loss, but some of these recombinants did. So they have increased pathogenicity. So this grant was never funded. Yet it has become a political hot topic because the politicians, particularly Republicans for some reason, will point to this and they will say, look, SARS-CoV-2 was made in the lab in China. They think this means that. They say they were doing gain of function, which this is, and they say this shows that the virus is made, which is completely ridiculous conclusion, of course. It was not made in a lab. It came from nature, but when you have people not understanding what's going on, that's what happens. And so now this experiment is why no NIH grant funds go to China anymore, because they think this was bad stuff. Why? Well, I have two theories. One is that they don't understand it at all. And the second is it's politically advantageous for them to do that. And you can decide which one, or maybe it's both actually. But the upside is that now gain of function is in the headlines. Risky gain of function studies. So all of a sudden, people doing gain of function are doing risky gain of function, when I just told you it's not the goal. Fight over COVID origins renews debate on risks of lab work. Talk of gain of function research. A muddy category at best. No, it's not muddy at all. It's very precise. I just showed it to you on one slide. Was that muddy? New evidence, US-funded gain of function research in Wuhan. Yeah, we knew they were doing this. This is the gain of function experiment. What's the big deal? Anyway, you see Republicans spin NIH letter about coronavirus gain of function research. Well, the consequence of this now is that in the current administration, an executive order was released in May, improving the safety and security of biological research. So this is a long document that says, dangerous gain of function research on biological agents. So if you talk to a politician now, every one of them will say gain of function is bad. That's what it means to them. They don't understand that it's more than that. But now it's got a bad rap. Has the potential to significantly endanger the lives of Americans. If left unrestricted, its effects can include widespread mortality, impaired public health system, et cetera, et cetera. This has never happened, by the way, right? I just want to say that. It's never happened. But I agree that we should regulate it, but not to the extent that this document does. Look at this. The Biden administration allowed dangerous gain of function research within the US with insufficient levels of oversight. This is a lie. This is a lie, because I know what was going on, and it wasn't dangerous, and there was plenty of oversight. I'm on a committee that oversees this kind of research. And you see here, through funding in China and other countries where there's limited US oversight. This recklessness, that's a politicization of this, calling it reckless. If unaddressed, may lead to proliferation of research on pathogens in setting without adequate safeguards, even after COVID-19 revealed the risk of such practices. So they are concluding here that COVID-19, SARS-CoV-2, came from gain of function research in China, which is of course not true. But if you go to covid.gov, you will see SARS-CoV-2 came from China, lab leak. And this is just unacceptable to scientists. And you need to understand this. Political leanings, whatever, this is not correct. So we have now regulations that are very onerous. They are not just oversight. So in the past, we had nuanced scientific oversight. Now it's just, you can't do these experiments. Anything that's deemed to be dangerous gain of function, your grant is terminated. And you can't fund any such work abroad either. So it's not an advisory role, it's just, you're finished. But what is dangerous gain of function? They say it's anything that will increase the pathogenicity, the transmissibility, the stability or immune evasion abilities of a human pathogen. So nobody actually does this with a human pathogen. So I'm not sure that anything is going to be prohibited, but that's what is in there, and there's just no need for it. I can't really talk much more about that, but I just wanted to give you a sense of what's going on. So scientists like myself object to this term dangerous gain of function. I mean, it conflates biology, routine biology, with risk. It obscures that most gain of function experiments actually reduce virulence and narrow host range. It creates confusion in public discourse. If you go out on the street and ask someone what's gain of function, they'll tell you it's making dangerous viruses. It's not. And we use gain of function to describe what an experiment does. Policymakers just look at it and say whether it should be restricted or not. So we are now in an era where the tools of science have led us to a conflict with policymakers that's going to have a long-lasting impact on science for years to come. All right, next time we will look at how viruses are built. We're going to talk about the structure of viruses.